go. Hmm. 1670. You guys see. Let's flip to share. Okay, so today we're doing infectious diseases, all right, of the skin. And then in lab, we're going to move into another set of infectious diseases. So this is the first time that we're actually going to take a body system and then talk about what it normally does, and then talk about the types of diseases that can come from that particular body system. And so we start with the skin because that's the first thing you see when you look at somebody, right? So we're going to describe the skin and talk about its natural defenses, and then we're going to get into the types of diseases that can impact it. Okay, so the integument forms the boundary between us and the environment. There we go. Okay. And it's formed by the skin, the hair, the nails, the sweat, and the oil glands. Okay, so what you're talking about here is a physical, chemical, and even a biological barrier. If we didn't have skin, we'd get infected much more frequently and much more easily. All right. And that would simply be because of the number of microbes that would be able to get into the deeper tissues and into the blood. And then even with a functioning immune system, they wouldn't be able to hold that off. Okay. The skin has a surface area of about 2 square meters and a thickness that varies from a few millimeters at the eyelids to 4 millimeters on the soles of the feet. And the skin is able to keep us pathogen free because it's constantly being shed in the upper layers. As dead cells get shed into the environment, the microbes that are on those cells go with them. And that means that the pathogens that are in there don't really have a chance to set up camp very effectively because we're constantly shedding them off and shedding them off and shedding them off. In addition, the flushing action of the sweat and the oil glands helps to keep the number of pathogens low, in addition to the fact that the normal flora that grow on the skin crowd out those bad actors. Okay. So that's just in the first initial layer, okay, that we have those defenses. So that's the epidermis, right? We replace it every month to month and a half, all right? The keratin in those cells allows them to withstand damage, abrasion, and water penetration. So it's like a wetsuit for your body. And that's to ensure that our fluid balance is maintained. If our skin were like a sponge, then we would soak up any water in the atmosphere or when we took a shower or when we got wet, and that would destroy our fluid balance. So the only way that water gets into our body is through our digestive system. The stratum basale is the stem cell layer. That's what generates all the cells above it. And as they migrate to the surface, they lose contact with the blood supply in the dermis, and they begin the process of apoptosis. And after that, they get shed. Okay. So this is a design to keep us pathogen free. Okay. If you damage the skin below the stratum basale, what you'll get is a scar, and that will heal with scar tissue, and that will be distinct from normal skin because it won't have any hair follicles or sweat glands or oil glands. Okay. But it will form a seal. Okay. The dermis is below the epidermis, and it's made up of fibroblasts and a lot of extracellular matrix with molecules in it like collagen and keratin and elastin, okay, that provide the toughness to the skin. And then wandering through that matrix are white blood cells, such as macrophages and lymphocytes and granulocytes. And they're looking out for anything that penetrates the epidermal layer so that they can intercept it before it gets into the blood. Okay. There's a lot of nerves and blood vessels and lymphatics that run through here as well. Damage that's deep enough to penetrate the dermis will break blood vessels. You'll bleed. Okay. Sometimes you'll get blisters, which are the result of a separation of epidermis and dermis because of friction. And note that follicles are roots of hairs that are located in the dermis. Hold on just one second.
I can't hear anything.
He said we'll be back.
I'm on hold for a second. Hold on. Okay. So that's your dermis. Okay. You might know the dermis better as leather, right? What you do when you make leather is you strip off the epidermal layer and you skin the animal and then you cure it, right? You know, leather is pretty tough stuff. So that speaks to the strength of your skin, right? Okay. Oil glands make the oils that create an antimicrobial sheen across the skin, okay? And they also flush the surface of bacteria. And the sweat glands do the same thing in terms of flushing, but they have a high salt content, and that prevents a lot of microbes from growing because it's very few microbes that like a high salt environment. All these glands are openings on the surface of the skin that are going to terminate in the dermal layer, but they are actually from the epidermis. Okay, they're surrounded by epidermal cells. And then we talked about the fact that the skin is being sloughed off constantly. Okay, so there's your skin. And what you want to notice, all right, let's get the pen out. Okay, is so the corneum is the dead part. So dead and shed. Okay. And then everything from here on down, let's use a different color. Everything from here on down is living in the epidermal layer. And of course, all of this is living material too. Okay. But notice that as the cells divide in this layer and move up towards the surface, they get to a point here in this living layer. This is called the stratum granulosum. That they form a barrier that's so packed with lipid soluble material that it's very tough for the diffusible elements of blood that the cells need to live to get up there, right? They get blocked. So those big things like oxygen and nutrients and so on. And so after that point, right, they die, right? This is all dead. And with them go the microbes that are living on and in that, okay? And that helps to keep us from infection. Notice these are oil glands, okay? Let me get a different color here, blue. Okay. Okay. Sebaceous glands. Okay. And then these are sweat glands. Okay. And they'll pipe their material up to the surface, right? And that'll help flush stuff off, right? You can see it in the hair follicles with the oils, right? That'll flush stuff off and that'll create an environment that most of the microbes really hate, okay? And that cuts down on the numbers of pathogens that can get on or in you. And then all throughout here, all throughout the dermal layer are immune cells, okay? White blood cells patrol the area. And if they see anything that looks suspicious, they're going to move in and eat it and kill it before it gets a chance to really get into the blood vessels, okay? So without your skin, you're in big trouble. In fact, people that have big time burns, one of the major problems that they deal with in burn units is massive infection. And that's not a surprise. So there are antimicrobial peptides. These are just proteins that are in the oils and the sweat that keep bacteria from reproducing, okay? The low pH of the oils of the skin makes it less likely that bacteria are going to grow. And that's because of the high concentration of lipid in the oil. The lipids are nutrients to microbiota, but the toxic byproducts are going to inhibit the growth of microbes that don't normally grow on the skin. So you better dig the skin if you're a microbe in order to be able to grow there, okay? So this makes a very unique, high salt, high lipid environment. There are components in the sweat that produce a low pH, as well as all the salt that's in your sweat that creates conditions in which a lot of bacteria don't like to grow. And then there's lysozyme, which attacks bacterial cell walls, which are in our sweat, our milk, our tears, and our saliva. And that makes the bacteria rupture if they get to an area that has a low solute concentration. A hypotonic environment will make them burst. Right? Let's write that down. Burst in a hypotonic. Stop it. Environment. Normally they wouldn't, right? But if their cell walls weak, they'll just pop like little balloons. Okay, talk about our normal skin bacteria, right? The stuff that doesn't cause disease, okay? And if we were doing this course on campus, this would actually be a lab that you would do using blood agar, right? So let's do right for blood agar. So you'd have a plate with blood agar on it. It's red, and it's opaque, right? Light doesn't pass through it. This is blood, right? In the agar, okay? So blood agar. Okay. And then what you do is you take a sample from your skin, say the palm of your hand, right? So you could say touch a part of the plate with your finger and then spread it with a loop. And then you'd let that grow for a few days in the incubator, 37 degrees. Okay. And then what you'd see when you came back would be little zones around some of the colonies that are perfectly clear, and then some zones that are kind of not too clear, kind of fuzzy, right? And then some colonies with no zones around them at all. Let's make the colonies, I don't know, make them gray, I guess. Yeah, so the colonies would be in the middle here. The zones of clearing will be outside. Okay, and the clearness of the zone and the size of the zone indicates to you whether it's a pathogenic form of staph called Staph aureus or a non-pathogenic form of staph that's called Staph epidermis. Okay, and the way that you classify that is by something called the hemolysis on the plate. All right, so if it's alpha hemolytic, okay, then what you got is a zone of clearing that's not quite so intense, and beta hemolytic is going to be a very intense zone of clearing and usually indicates the presence of Staph aureus. Okay, the color of the colony will help as well, because there's stuff in the blood agar that the Staph aureus can use for fuel that the Staph epidermis can't, and that'll turn the colony a certain color in some cases. Okay. But this is what you get with a blood agar plate. It's made with agar and with sheep's blood. Okay. And the reason that this indicates the presence of the pathogen is that the enzyme that does the lysing, all right, is part of the bacterial strategy to get iron out of the blood to use for growth, right? And for those bacteria that are pathogenic, they have a tremendous ability to rupture those red blood cells. And for those that aren't, they don't so much, right? So the epidermis would either have alpha or no hemolysis, and the aureus would have beta hemolysis, which would be seriously cleared zones. Okay. So in order to live on the skin, you've got to dig dry and salty conditions. The bacteria there grow in dense populations in areas of the skin that are moist, and that makes sense because water promotes life. They also live in hair follicles and glandular ducts. This is, for instance, where we get acne. Okay. The Human Microbiome Project was just a way to determine how many and what kinds of bacteria were growing on your body and in your body. And they found that there were hundreds of species, including a lot of pathogens, that are in the epidermis, the dermis, and even in the subcutaneous layers, with different species and different parts of the skin and different parts of the body. And even different people have different species of bacteria growing on it. Okay? But whatever it is you've got tends to stay relatively constant in terms of composition over time. Right? So there's some sort of interaction between you and your genes and what can grow on you easily. Okay. So we'll talk about MRSA. We'll talk about treatments for macropapular rash diseases. We'll talk about cellulitis, scalded skin syndrome, and some other different diseases. And we'll talk about mycoses, which are caused by fungus. Okay. And then rubella and rubeola. Okay. So macropapular rash diseases are skin eruptions that are caused by microbes. Okay. They generate flat to slightly raised colored bumps. Okay. And let's here. We knocked over a bunch of stuff. Hold on. Let's hold on a second here. Okay, so I'll do this. And then we want to look down this and unhide. There we go. Okay. All right. So MRSA is an acronym. Okay. And you probably heard the term MRSA before if you've ever been in a clinic or in a hospital. It's one of those infections that we call nosocomial, meaning you get it in a clinical environment. Let's write that down. From a clinical environment. Okay. And MRSA stands for methicillin resistant Staph aureus. Okay, so that's the letters of the acronym, right? MRSA, MRSA. Okay. So it causes skin lesions in people that aren't even hospitalized. The strains are resistant to antibiotics that are from the penicillin family. That's what the methicillin is. Okay. But there's other antibiotics it's resistant to as well. It's a gram-positive coccus. Okay. So that means it's going to be on a gram stain, look like purple clusters of round cells. Okay. It can be highly virulent, meaning it can cause disease very easily. But it's also found in normal skin in a third of the population. Okay, so most of us have a little bit of it on us, but it gets crowded out by the staph epidermis. It can withstand up to 10% salt, extreme pH, and high temperatures. It is still alive after it's air dried, and it can resist a lot of disinfectants and antibiotics. So you see a red, tender, localized lesion, like in the panel below. It has pus and it feels warm to the touch. It can occur in breaks in the skin and may form around a hair follicle. A fever may be present in the area. It'll get warm. Okay. How do you get it? Well, from any surface, really. I mean, Staph aureus is all over the place, including on you right now. Okay. But it likes breaks in the skin. Okay. Persons with active MRSA should keep these areas covered so that they don't transmit it to other people. All right. So what is it that makes it so bad, right? What has coagulase, right? Coagulase is an enzyme that dissolves clots. Okay. It also makes hyaluronidase, staphylokinase, DNase, and lipase. And what these do is they help to break down the dermis. There. Break down the dermis. Okay. Okay. And use lipid for food. Okay. We can determine its presence using PCR, which is basically DNA analysis. Like the blood work they do at crime scenes. Okay, same idea. We isolate on blood agar or mannitol salt, right? The blood agar, because that's the environment it's trying to get to, right? Mannitol salt, because it's going to grow on that. And at the same time, other bacteria won't be able to because it's got a lot of salt in it. The catalase distinguishes the Staph aureus from other staph, okay? And so what that means is that it will bubble with hydrogen peroxide. okay? And you could do a coagulase test for it as well, where you throw it in with a clot that dissolves the clot. Prevention, good hygiene, right? Wash yourself. Keep the numbers low. Treatment through cutting out the lesion if it gets real bad and draining the pus, and any antimicrobials, clindamycin, or doxycycline, okay? So you can see here how this works, right? You want to figure out whether or not you've got the staph infection. The test tube's got plasma in it, all right? We inoculate the plasma with staph, all right? And a positive test, all right, would be little clumps forming, all right? And then dissolving, right? A negative test would be no clumps forming. So that's the breakdown, right? What causes it, how it's transmitted, the virulence factors, right? This is these are weapons that make it a pathogen. Okay, that's how we analyze whether it's there. It's how we prevent it. Stay clean. That's how we treat. Okay, and then epidemiology is where it is spread. Okay. Now you guys are going to do like little mini reports in the coming weeks about diseases that I'll pick and give to you. And this is basically the form I want the report to take, right? You tell me, I'll give you a disease. You tell me what causes it, how it's transmitted, what makes that organism make you sick? What is it? What are its weapons? Okay. And then how do you prevent it? And how do you treat it? Okay. So I want this and this and this and this in the report. Okay. And it's just like a 1-page deal, just bang, 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 bang. Okay. Okay. Macropapular rash disease are eruptions that are caused by different microbes that generate colored bumps. All right. And one of these diseases is called measles. Okay. So let's take a listen to measles.
Cram. Hi, welcome to another MedCram lecture. We're going to talk about measles. There's been an outbreak of measles recently, so I wanted to talk about the clinical syndromes that measles can cause, the stages of infection, the diagnosis, and the treatment. And I think there's some high-yield things that any healthcare provider should know. First thing you should know is that it's a virus, okay? Specifically, it's the rubeola virus. Rubeola. Now, you may remember the MMR vaccine. This is measles, mumps, rubella. Rubella is German measles. It's a more milder type of disease. This is the first M, the measles, mumps, rubella, okay? And it's the rubeola virus. It's very infectious. In fact, the estimate is that if somebody has the virus during its transmissible phase, they can infect up to 12 to 18 people. So it's very infectious, as we've seen in the news, and it can spread very rapidly. Now, just to be complete, there are 6 different clinical syndromes that the rubeola or the measles virus can cause. Of course, the first one is the classic type of measles, otherwise known as classic measles. We're going to talk about that one more. That's the, your 3 C's, which we'll get into, the Koplik spots, which you've heard about. We'll talk about that one. Next one is called modified measles. Modified measles is where someone who has been immunized in the past, but maybe don't have a good antibody response, may get a sort of a lower level classic measles. It's called modified measles. Usually the onset is longer and the course is not as severe as would be with classic measles. So yeah, it's possible that you could be immunized against measles and still get a measles type of syndrome, but it's a little bit less. Third one is called atypical measles. We don't see it much anymore because it has to do with people who got the dead virus vaccine. Now, most measles, mumps, rubella, in fact, all of them are a live virus. That's why we don't give it to people who are immunocompromised. But the dead virus, which was given back in the mid-60s, would not give the proper immune response. And so it was possible for these people who were immunized with the dead virus could get this atypical measles, just something you should know about. A couple of other things, or 3 other things that you can get, is a post-infectious neurological symptoms. So post-infectious, neurological. Okay, these are some of the things that you might hear about called ADEM. That's acute disseminated encephalomyelitis. The other thing you may have heard about is SSPE, which is a subacute sclerosing panencephalitis. These are not good. These are complications. We'll talk about those briefly. The other one you can get is severe measles. I'm not going to talk much about that. And then other complications, such as things called giant cell pneumonia. PNA is my abbreviation for pneumonia. And then you can even get something called a measles inclusion body encephalitis. So these things here, basically 4, 5, 6, these are complications which we'll talk about briefly later. But the one that I want to focus in on is the most common, and that's the classic measles. How are you going to be able to identify that in patients that you see in the clinic, in the emergency room, etc.? So let's talk about that. There are 4 stages of the infection of the measles virus, and I've got them abbreviated here, I, P, E, and R. I stands for the incubation period, P stands for the prodrome, E stands for the exanthem, and R stands for recovery and immunity. So in the first phase, called the incubation period, so incubation, the patient is usually asymptomatic. They don't feel any symptoms. Very rarely will they feel symptoms. And the key here is that that can last from 8 to 10 days. Okay, so they're walking around for 8 to 10 days and they don't even know that they've been infected. Now, the prodrome is an interesting period. It's here where you see the 3 C's, prodrome. But the first thing that comes on is constitutional symptoms such as fever and malaise and anorexia. This doesn't mean that they're skinny, it just means that they don't want to eat. But the other thing that you see here that's very characteristic is something called the 3 C's. And what are those 3 C's? You ought to know what those 3 C's are. The 3 C's are cough, conjunctivitis. That means that the area around the eye is inflamed and it is red. And the last C is coryza. Basically, another name for coryza is rhinitis, or inflamed nasal mucosa. So runny nose, eyes that are kind of red, and cough. So these are kind of non-specific, but if you see those three together with fevers, especially when you're thinking about an outbreak, you want to think about that. Okay. The next thing you want to think about too is something that's very classic that they'll test you on in this prodrome phase, and those are Koplik spots. Now, what are Koplik spots? Some people have described this as grains of salt on a red background. If you were to look in someone's mouth, imagine the mouth is very inflamed and it looks red back there. And you can imagine either whitish, grayish, or bluish. So if I were to pick a color like that, you would see these whitish, grayish, or bluish spots right where the molars are on the buccal mucosa. They could also see it in other mucosal areas, genital specifically, but this is typically where you see these Koplik spots. Now, the interesting thing about Koplik spots is that they usually show up 48 hours before the next stage, which is the rash or the exanthem stage. Okay, Koplik spots are known as an enanthem. The rash is known as the exanthem. So if you see Koplik spots, the rash is probably going to be coming in within the next 48 hours. By the way, the incubation period we said lasts about 8 to 10 days. The prodrome period usually lasts between 2 and 8 days, but mostly it lasts about 2 days. So if it's lasting about 2 days, and Koplik spots usually pop up 48 hours before the next stage, usually the prodrome comes on with fevers, malaise, anorexia, cough, conjunctivitis, coryza, and Koplik spots. The thing you ought to know about Koplik spots is there's nothing else that causes them. If you see Koplik spots, it's what we call pathognomonic for measles. Okay, now let's talk about the rash. The rash is a maculopapular rash. Okay, and it starts on the head and moves down. It usually spares the palms and the soles, which is important to understand. Now, the other thing about this is that after the rash, so if the rash starts usually about 2 to 3 days after the rash, there will be a fever or high fever, which will then defervesce. And in fact, after the rash appears, usually clinical improvement occurs about 48 hours after the rash appears. So again, another 48 hours. So let's review that 48 hours. We have two 48-hour marks. 48 hours before the rash appears is when we'll see the Koplik spots. And 48 hours after the rash appears, we'll see fevers, but then they will start to go down. If we don't see resolution of symptoms after 48 hours of the rash, these people are at increased risk for getting complications. The other thing you should know is when the rash appears is usually when the Koplik spots disappear. Finally, let's talk about recovery. Usually this could last a couple of weeks, but eventually the patient is going to get better. The cough may persist for 1 or 2 weeks after the infection. But again, if you start to see fevers beyond 3, 4, 5 days after the rash, usually that may be leading toward a complication. So fever, 48 hours plus post-rash equals questionable complications occurring. Let's review the key portions here. There's 4 stages. The incubation period lasts for more than a week. When the prodrome comes, there's fever, malaise, anorexia, the 3 C's, Koplik spots appearing 48 hours before the rash, and then when the rash comes, the Koplik spots go away. They'll have fevers continuing. But after 2 to 3 days of the rash will then start to coalesce and then basically slough off, And then if fevers continue after 48 hours after the rash, that's usually not a good sign. Finally, the recovery phase is about 2 weeks. Great. In the next video, we're going to talk about the diagnosis and the treatment of measles.
Hello, my name is Dr. Sue McGladdery. I'm the Medical Director from First Med Centres in Budapest, Hungary. I'm going to give you a little bit of information about rubella. Rubella, or German measles, is a viral infection that causes a mild measles-like illness, so-called 3-day measles. But there is one important exception when pregnant women get rubella. In this case, the consequences for their unborn child may be very severe. Up to 85% of infants born to mothers who had rubella during the first 11 weeks of pregnancy develop something called the congenital rubella syndrome. This can cause one or more problems, including things like growth retardation, cataracts, deafness, congenital heart defects, and even defects in other organs. The highest risk to the fetus is during the first trimester, but exposure later in pregnancy can also be dangerous. There is a way to prevent rubella, and that is the measles, mumps, rubella vaccine. It's very effective, so that now rubella is very rare in most developed countries. However, cases of rubella do still occur, mostly in unvaccinated adults. Fortunately, cases of babies born with the congenital rubella syndrome are now very rare, thanks to the vaccination. So what can you do to prevent congenital rubella syndrome? Well, if you're planning to become pregnant, you should really check your vaccination record in advance to make sure that you've received all of your MMR inoculations. It's a good idea to see your doctor for a preconception visit. A simple blood test can confirm that you're already immune to rubella. It's best for women to be protected against rubella before pregnancy. So if you know you're not immune, get the vaccination before you become pregnant. If you are pregnant, most doctors will make a routine screening for immunity to rubella as part of the prenatal care program. The other thing that you can do, of course, is to ensure that your child receives the recommended 2 doses of the MMR vaccine on time.
Fifth disease, sometimes called erythema infectiosum. Okay, let's break that down. Okay, erythema means red. Okay, and infectiosum is referring to the fact that it's communicable. Fifth of the diseases recognized by doctors to cause rashes in children. That's why it gets the name fifth disease. Okay. So what are those diseases, right? Scarlet fever, measles, rubella, and another rash thought to be distinct. So what are the signs and symptoms? Mild disease that shows up as a slapped cheek appearance beginning in the face. In 2 days, the rash spreads on the body, but is mostly on the arms, the legs, and the trunk. And it can occur for several weeks and can be brought on by any activity that increases body heat. It's caused by parvovirus, okay? So we diagnose it by looking at the patient, right? You have that slap cheek redness. We rule out rubella by checking for antibodies against rubella, right? IgM, remember, are the first antibodies generated in an infection. So they're the first on the scene, but they're less effective than the IgGs, right? But they show up in the blood. They're big, right? Remember, they're pentamers of antibody. Transmission, it's very contagious. Prevention, no vaccine, no treatment. Okay. So, you get it, and then your body kicks it, right? It's generally a mild disease. So what you do is, what we call that is, it's self-limiting. Okay. Which means that it'll resolve on its own. Okay. So your body has to kick it, right? But once that happens, you're going to have memory cells, memory lymphocytes, and you shouldn't get it again, at least unless it mutates. Roseola, sometimes called sixth disease, generates a macropapular rash, but up to 70% of the cases can happen without a rash. Usually see a high fever that comes on quick and lasts 3 days. You may get seizures. On the 4th day, the fever vanishes, the rash appears on the chest and the trunk, and then a little bit less on the face and the limbs. It's caused by herpes virus 6 and sometimes 7. This can be latent indefinitely after the disease is cleared, okay, which means it can reemerge. Okay. Occasionally, reactivities in childhood or adulthood cause monosymptoms. 100% of the US population is affected with this by adulthood. So it's just here, okay? How do you prevent? No treatment, no vaccine, okay? So again, what we say is it's self-limiting. Resolves on its own. But because it can stay resident, it can recur. Okay. And there they are. There's a list of the different macropapular rash diseases, what causes them, what the virulence factors are, right? What is it about them that makes them cause disease? Okay. So, like, look at measles, right? You form syncytia, which are what? Fused cells, right? So the virus gets in and the cells fuse. Okay. It can suppress immunity. Okay. Rubella, it can inhibit mitosis. It can induce apoptosis, which is programmed cell death, and it can damage the blood vessel lining. Okay. And then here, roseola is that latency, the ability to just live inside your body as information in the nucleus until it wants to pop out again. Okay. So remember, these are the weapons. Okay. What are the treatments? Right? Nothing, right? We just got to let it resolve. There's no antivirals for measles. You can use vitamin A and antibiotics for what we call secondary bacterial infections. Now, what is that? That's what sets in after the virus has done its dirty work, right? Is the bacteria can follow it and cause another infection. So what you're doing is treating for that, not for the virus itself. Okay, remember that we talked about the fact that antivirals are tough to develop because viruses generally use our own machinery against us to reproduce. Okay. Okay. Impetigo. Let's take a listen.
Upper St. Francis Healthcare's House Calls TV.
Impetigo is a contagious skin infection, especially common in children because it's often spread at daycare or school. Dr. Valerie Scott describes impetigo. It'll start as a red bump or a little blister, and then it'll start to spread, and you'll see the top of it looks like honey-colored crust. It'll be very crusty and maybe a little bit red. Your child won't have a fever. Will basically be healthy, but the infection will continue to spread if they scratch it. Impetigo comes from bacteria like strep or staph and can be treated. What you can do is clean the area. You can soak the child's scab and remove it. I would probably wear gloves while doing that so that you don't get it yourself. If the crust keeps coming back or it spreads, you really do need to go see your doctor. The doctor will either treat you with a topical antibiotic that's by prescription or an oral antibiotic, depending on how widespread the infection has become. For more information, call 402-CARE. For House Calls TV, I'm Mara Dempsey.
Upper St. Francis Healthcare's House Calls TV.
So if you look, impetigo causative agent here, Staph aureus or Staph pyogenes. Okay, so these are staph infections. All right, so impetigo is a bacterial infection. Okay, so that's different from rubella, roseola, and all the stuff we've been looking at up to this point, right? The treatment as a result is going to be antibiotics, all right? Antibiotics, right? And what is it that makes it cause disease, right? Well, for Staph aureus, it's something called exfoliative toxin A. What is that? It's a protein that causes skin shedding. Okay. It also has a coagulase. Okay. And there's other enzymes that can break through extracellular matrix, right? Over here, strep pyogenes. It can bind plasminogen, okay, which means that it can prevent blood clotting. And it has hyaluronidase that lets it eat through extracellular matrix, okay? And it has proteins that produce an inflammatory response, okay? So those are the weapons. Those are the weapons. Okay. Okay. Let's take a look at cellulitis.
Hi, I'm Dr. Enoch Choi of medhelp.org, and I wanted to share some helpful tips on how to manage cellulitis, a skin infection that sometimes requires antibiotics. The main thing to remember with a skin infection is that you need to keep that area of your body that's infected elevated over the level of your heart. The reason why that is that it helps drain the infection away, allows your body to be able to attack the infection with its white blood cells, the cells used to attack infection. and it helps deliver the antibiotics better because that area isn't so swollen, and the blood supply can deliver the medication that you're taking. It's important to remember that if you get a high fever, if you have shortness of breath, if you have any worsening before you're getting rechecked, it's something that you need to talk to your doctor about and consider being seen for. It's pretty hard to work when you have a skin infection like this, since you need to keep the area of infection above your heart, so you probably won't be able to work. You know, it's something that it's hard enough to do that. Oftentimes you'll need to probably stay on the couch or just be resting with that part of your body, if it's an arm or a leg or whichever part of your body elevated above the level of your heart. And you want to do that all the time, and probably for at least for the next couple days. You probably can't do your regular work that way. You want to keep if there's a wound, if there's an opening in your skin, you want to keep that clean and dry, but you can take showers. You want to change bandages over this area every day. And if you have been told by your doctor to do so, you can use some polysporin to try and prevent any local infection of the opening, and also keep the dressing from sticking. That makes it a lot more comfortable to take the bandage off. Oftentimes your doctor will ask you to come back for a recheck. If so, you should probably go and do that. And sometimes that's something that's done on a daily basis, especially if you need another dose of IV antibiotics, which some people need if the infection is serious enough. Now, how are you supposed to manage just lying on the couch all day? Well, you can take bathroom breaks. Try it, make sure you can take a short walk, like for 10 minutes, say 3 times a day, and ask somebody to help you, or you may need a cane or a walker to do that. And when you're not on that walk, keep that area elevated. I haven't told you enough times now, you have to keep it elevated over the level of your heart. That'll let the swelling go down. If you get the area wet with a shower, make sure you pat it dry after washing it. Put a clean dressing, a bandage, a Band-Aid on it after you wash. Whether or not you're showering, you can change that every day. Try and put something warm on that area, on the red part, every day, say 4 times a day for about 10 minutes each time. And that can help deliver more of your blood flow to the area. And make sure that you don't put anything restrictive, any tight clothing above the area that's infected, because you don't want to block blood flow to the area. These are some helpful tips, and of course you need to ask your doctor about specific instructions, but they're helpful hints for dealing with a skin infection called cellulitis.
So you can see here some of the different microbes that cause cellulitis. Okay, again, they're mostly Staph aureus and Pyogenes, right? And then there are some fungi that can cause this as well. And again, the weapons are the same, right? There's that exfoliative toxin, right? There's the coagulase. And over here, you've got the plasminogen binding activity. It's going to prevent blood clotting. Got the hyaluronidase. It's going to allow it to drill through dermis, okay? And the streptokinase, it can promote inflammation, right? So those again are the weapons. Okay. And how do you treat? Again, antibiotics. Okay. Sometimes you might have to go to surgery, all right, in order to prevent it from spreading into the blood. But that depends again on the type of infection that you have. Often this is going to come through breaks in the skin. Okay, such as a cut or a puncture. And so where are these pathogens? They're always there. Okay, they're in the environment, they're on you, they're in you. They're just kept in check by our defenses, right? And our normal bacteria. But sometimes with a break in the skin, you get unlucky and you get one of these pathogens to get a foothold and start to grow. Okay, scalded skin syndrome is produced by a strain of Staph aureus that contains genetic material for exfoliative toxin. Okay. Okay. So again, that's that same weapon, right? It causes excessive skin shedding. It's a dermolytic condition, can hit newborns and can hit adults. You get lesions because you get a split in epidermal tissue above the germinal layer. It first appears around the umbilicus. That's where your belly button is, your navel, okay, in neonates, or in the axilla, which is your underarms, right? Armpit region. Okay. You see widespread skin shedding, all right? How do you transmit? Often when the caregivers are in contact with the baby or in contact with multiple babies and spread it from one baby to another. Adults can do this. 30% of adults are what are called asymptomatic carriers, meaning what? Show no symptoms. Okay. And they can have the organism in the nasopharynx, which is the part of the throat in the back of the nose, the axilla, which are the armpits, the perineum, which is the genital area, and the vagina. Okay. Diagnosis by looking at the patient. Okay. Prevention, get the carriers out of contact with the children and treat with antibiotics. All right. So there it is, scalded skin syndrome. Okay. And there's the weapon. Okay. All right. Pustular diseases. Chicken pox is very common and often benign. Smallpox is dangerous, right? A single case is a public health emergency, and both are generalized rashes. Pox is just a standard term meaning individual lesions that contain fluid. Okay. So let's take a break at this point and let's come back in 15 and we'll take the second half of this on. Okay. So cut here. We'll take a break. And I'll see you guys back here at quarter after. Okay. Here's a little bit on chickenpox.
Post-herpetic neuralgia, in contrast to diabetic neuropathy, most commonly manifests as mononeuropathy, meaning the condition affects a single nerve root. The herpes zoster virus causes chickenpox, but also remains dormant in the body's dorsal root ganglion cells once chickenpox has subsided. The herpes zoster virus can reactivate at some later point, causing shingles. Why the herpes zoster virus reactivates in a specific person is rarely understood. However, known causes include immune system depression and other CNS conditions, including stress, hormonal changes, or nerve injury. When herpes zoster reactivates, it inflames dorsal root ganglion and infiltrates dorsal root neurons. This inflammation hinders dorsal horn cellular metabolism. In particular, it impairs nerve conduction and pain transmission. The herpes zoster also targets the peripheral segment of the dorsal root ganglion. The virus travels from the dorsal root ganglion to peripheral nerves, causing shingles characteristic rash. Once in the peripheral nerves, herpes zoster can cause peripheral demyelination and fibrosis.
So you can see here an example of the nerve roots on the back of the body. These are the cervical division, the thoracic, and the lumbar. And what you can note on this individual is a latent infection that has gone from lysogenic to lytic, right? And now we have virus production, and the virus is emerging from the nerves that are fed by the roots from C7 through T1, right? And it's going to wrap all the way down, and it's going to follow even the nerve pathways that branch off of those nerves, right? So you can see very clearly the dermatome here, all right? And that's what causes the pain, is the eruption of the virus from the nerve endings. So how do you prevent this? There was a live attenuated vaccine that was licensed in 2006. Okay, so again, let's break that down. Okay, attenuated means not capable of infection. Okay, so they've taken the virus, they've treated it so it can't infect, but you can still make antigen to it through the vaccine, right? And we have Zostafax that was approved in 2006 for the prevention of shingles, okay? Varicella is self-limiting, right? Meaning what? Well, we talked about that already. It resolves on its own. Okay. And the only therapy is to deal with pain. Okay. You can get secondary bacterial infections, and you just use antibiotics for those. Okay. A lot of viral infections, the way it works is that the primary infection is viral. Okay. And the secondary infection is bacteria. Okay. Okay. This we can treat. Okay. this, the treatment is limited. Okay. Oral acyclovir is given to those considered at risk for complications. So this is actually an antiviral. Okay. So it cuts down on the virus's ability to reproduce, but it's not 100%. But in combination with the antibiotics, the idea is to limit the primary tissue damage and eliminate the possibility of a secondary infection. Okay. Smallpox, very dangerous disease, right? Smallpox is a disease of the past as a result of actions of the WHO. There was some anthrax bioterrorism in 2001, and vaccination had been discontinued, but now we offer it to US populations thought to be at risk. So what are the signs and symptoms? Fever, malaise, a rash that begins in the throat, spreads to the face. The rash is macular, papular, vesicular, and pustular, and crusts over and generates non-pigmented sites with scar tissue. Variola major is the highly virulent form that causes toxemia, shock, and intravascular coagulation. It's important to recognize the signs and symptoms. This is a law enforcement emergency if you even have one case. The prodrome, it shows high fever and malaise, okay? The rash emerges in the mouth and you get abdominal and back pain. The rash spreads all over the body in 24 hours. After about the 4th day of rash, bumps get larger and fill with the thick, pussy fluid. Pustules are indented and scab over in a few days, and after a couple of weeks, the lesions have crusted over, but the patient is contagious until the scabs fall off. During the rash, the patient is very sick. The lesions heal on the dermal level, so scars are left behind. In variola minor, the rash is less dense, the patient is less ill. The variola virus is the agent of infection, right? This is the pathogen. It's an envelope DNA virus, which is a member of a group that includes monkeypox and vaccinia. It is real tough. It can survive outside the host longer than most viruses can. It's spread through droplets. Fomites can be contaminated. Bedding and clothing can spread it. It was endemic in 31 countries in the 1970s, and about 15 million got the disease annually, with 2 million dying annually. A vaccination effort that lasted over a decade got rid of the disease, and the last case was in 77 in Somalia. The treatment, no treatment, okay? No treatment. So for viral diseases, you're going to generally find this, viral diseases require vaccination. Since treatment is limited. Okay. So you can see the difference between the two, right? There's your chicken pox, okay? There's your smallpox, right? What are the weapons? Again, the ability to fuse cells and remain latent, right? So once you get chicken pox, you've got it for the rest of your life. And here, It can avoid the immune response. Okay. Avoid the immune response. So it has the ability to evade white blood cells, or to make it difficult for them to even detect the virus, because remember that this is an enveloped virus. He's disguised, right, in host membrane, and so that makes him difficult to detect. Okay, leishmaniasis. It's a localized infection of the capillaries in the skin. In mucocutaneous leishmaniasis, we normally see it in Central and South America, and it affects both skin and mucous membranes and can cause systemic leishmaniasis. It's caused by Leishmania tropica, okay? Whereas mucocutaneous is Leishmania silencius, okay? So two different protozoans, okay? The zoonosis transmitted among mammalian hosts by female sandflies. It's endemic to equatorial regions that provide conditions for the sandfly to live. So travelers and immigrants are at risk. Okay, so these are protozoa, right? These are not bacteria. These are not viruses. Protozoa. These are eukaryotes. Okay. We diagnose it by culturing and seeing them under a microscope slide. No vaccine. Avoiding the sandfly is the only prevention. Okay. So the sandfly is the vector. Okay. That is the vector. All right. Otherwise, you're going to get the disease, it's going to cause its damage, and then your immune system will eventually get rid of it. Okay. In cutaneous anthrax, you get papules on the skin that get necrotic, meaning what? They die. And later rupture to form a black eschar. It's like a scar. The causative agent is Bacillus anthracis. Transmission and epidemiology. The endospores enter the skin through cuts or abrasions. Okay, remember that endospores are dormant. Bacterial seeds. I put that in quotes. Okay. Male workers and others can get the infection when endospores get sent through the mail. It can be naturally transmitted by contact with hides of infected animals. We culture it on blood agar. And what do we look for? We look for hemolysis. Okay. We do serology, and that involves the use of antibodies to determine what strain of bacillus you have, right? There's PCR that you can do, DNA analysis as well. If left untreated, cutaneous form is fatal in 20%. There's a vaccine, but it's only recommended for high-risk people and the military. Okay, so there is a vaccine. Okay. So you can see here, two types, right? This is a protozoan. And this is bacteria. Bacteria. Okay. Sandfly is the vector here. Okay. Direct contact is the vector here. Okay. The mode of transmission. The virulence factors. Well, for leishmania, the protozoan can reproduce inside the white blood cells, the macrophages, right? For cutaneous anthrax, there is something called lethal factor and edema factor that produce the disease state, okay? The endospore is a way to survive and get transmitted, okay? And get in the first place, all right? Treatment. Sodium sibgluconate and pentamidine. What are these? These are anti-protozoals. Okay. And these, of course, over here, these are antibiotics. Okay. All right. Ringworm. This is caused by a fungus, okay? Dermatophytes are a group of fungi that cause different body surface conditions. It's confined to non-living epidermal tissue and their derivatives, the hair and the nails. So these are all dead. Eraser. Dead, stratified, squamous, epithelia. Okay. Epithelia. Right. So this is the fungal food. Okay. All conditions have names that begin with tinea, derived from the erroneous belief that they were caused by worms. They're not caused by worms. They are caused by fungus. Fungus. Okay. So let's take a look at ringworm.
Hi, I'm Dr. David Hill, and today we're going to talk about the best ways to disinfect for ringworm. The first and most important thing to know about ringworm is it is not a worm. It is a fungal infection of the skin. It seems because of the round shape, people used to think maybe it was caused by a worm or was a worm, but it's not. It's a fungus. Now, it is transmitted both from skin to skin, so people who engage in sports like wrestling or football that involves skin-to-skin contact are prone to get it. It can also be transmitted on clothing, on towels, and on surfaces like locker room, benches, and floors. So if it's possible to clean those surfaces, especially if you know somebody using them has had ringworm, you want to do so. Now, the same 10% Clorox solution that you could use to clean almost any surface will work beautifully for surfaces that are not damaged by Clorox. If you're looking at a fabric, for example, you probably want to spot test it before you clean with that. There are special detergents that can be used to kill fungus that grow in clothing or towels. I'd also recommend a hot water cycle. And again, if it's something that's color fast, you might try bleach. On your own body, you want to get rid of the ringworm using an antifungal medicine. And any of the athlete's foot or jock edge creams that are sold over the counter in the drugstore work just as well as anything that we have a prescription to give you for. The key is to keep treating for a week after the rash disappears. That's the hard part, because once the rash has gone away, you kind of forget about it. But you want to keep that cream out there where you can remember to keep putting it where the rash was. You also want to treat beyond the margins of the rash by about an inch, because the fungus is out there even though you don't see a skin reaction yet. So remember, ringworm is a fungus. You do want to disinfect potentially exposed surfaces, ideally with a 10% Clorox solution. And you do want to wash potentially exposed clothing in hot water, laundry detergent, and even bleach if possible. Talking about disinfecting for ringworm, I'm Dr. David Hill.
So these are what are called superficial mycoses, meaning fungal infections that happen on the surface of the body. Okay. They're caused by a genus of yeast that has at least 10 species that are already on the skin, and they eat the oil on the skin, and they also eat the dead skin cells, right? They also may cause folliculitis, psoriasis, and seborrheic dermatitis. There may be contributing factors to that. Everybody has this, pretty much. Everybody has these on them or in them, okay? And so you can see here, again, if we look at the chart, right? The organism, right? The organism is a fungus, okay? Funguses live off dead tissue. Okay. For cutaneous, those are the fungi. For superficial infections, these are the fungi, right? Again, fungus, right? So how does it cause disease? Well, it can break down keratin and cause inflammation, okay? Hypersensitivity is like allergy, okay? And the keratin is part of the dermis, right? So it's literally tearing that stuff up, right? For superficial infections, okay, we are feeding on epidermis. Okay. We diagnose it by looking at a sample, okay? The treatment, all of these, okay, are antifungals. Okay, different from an antibiotic and from an antiprotozoal or an antiviral, right? Here, they say topical antifungal. Topical means applied to the surface. Okay. So these guys are everywhere, all right? They're everywhere. Okay, we'll talk about the eye, all right, and list some natural defenses, all right. So the eye is the organ of vision, right? And there is a membrane-like tissue called the conjunctiva that lines the eyelids and has a lot of blood vessels in it and makes oil and mucus that lubricates and protects the surface of the eye, along with the production of tears by the lacrimal glands that are generally found up here in the superior lateral margins, lacrimal glands. Okay. And the idea is that the tears wash the eye and contain lysozyme and keep the eye moist, so it keeps the cornea from drying out. But it also flushes away pathogens. And also the moistness allows the diffusion of oxygen into the cornea because the cornea has no blood vessels. Has no blood supply. So it has to have direct contact of a moist surface with the air, right? The cornea is the dome-shaped portion of the eye that lies over the iris, and the epithelial cells regenerate quickly if they're damaged, all right? And the epithelial cells here are transparent. Okay. So light, when it hits it, just bends instead of scattering, okay? So we sometimes nickname this the windshield of the eye, right? Tears contain water, oil, mucus, and things like lysozyme and antibody, okay? They're formed in the lacrimal glands shown here, right? That's the lacrimal gland, right? And they drain into the lacrimal duct at the inner corner, and then into the lacrimal sac and down the nasolacrimal canal into the nose, okay? That's the flow of tears. Lactoferrin is also found in tears, okay? And lactoferrin is simply a protein that binds iron, okay? And the mucus layer has proteins and sugars in it, okay? The flow of tear film is going to prevent the attachment of microbes to the eye surface and provide a moist surface so that oxygen can diffuse into the cornea and keep those cells alive. The function of the eye is vision. Inflammation doesn't occur in the eye easily as it does in other parts of the body. If you flood the eye with light diffracting objects like lymphocytes and phagocytes, your vision would blur, right? So there are 4 transparent layers that light has to pass through to get to the back of the eyeball, which is your retina, right? And that's the cornea. And the aqueous humor. Okay. Then the lens. And then the vitreous humor. Okay. All transparent. Okay. So the cornea of the eye has what we call immune privilege, meaning that if you wanted to transplant a new cornea into an eye where the cornea had been damaged, you could do that without fear of rejection. And that's because there's no blood supply. The oxygen comes from, again, the diffusion of the oxygen from the air directly into the eye tissue. Okay? So what kind of microbes do we find on and around the eye? Okay, not many, right? 20% of people tested have no bacteria in their eye. The bacteria found in the eye, if they find it, is usually staph or strep or cornybacterium, occasionally yeast or Neisseria, all right? But generally, the eye is microbe-free, okay, under normal conditions. So we're going to talk about conjunctivitis and its prevention, treatment, and so on. And then we'll talk about keratitis caused by herpes simplex or acanthamoeba, okay? So conjunctivitis is a bacterial infection of the conjunctiva, all right? The itis literally means inflammation. Okay. Contaminants proliferate because of the presence of a contact lens if you use those, or you can injure the eye and bacteria can get a foothold, or you can accidentally get bacteria in your eye through trauma, all right? Like poking a finger in somebody's eye. So what do we see? A milky discharge, whereas viral infections make a clear discharge. Patients wake up with the eye glued shut by secretions that have accumulated overnight. Some conjunctivitis cases are caused by allergic responsive, and you have a clear discharge in that case. It's commonly called pink eye, all right? And that comes from the inflammation of the blood vessels that run through the conjunctiva. Neonatal eye infections can be caused by Neisseria, chlamydia, and be transmitted vertically from the mother to the child, okay? It can lead to serious eye damage if you don't treat it right away. Herpes simplex can cause neonatal conjunctivitis, but is usually accompanied by a herpes infection elsewhere in the body. So this would be like getting chicken pox in your eye, okay? Bacterial conjunctivitis is caused by staph epidermis, okay? Strep pyogenes, strep pneumonia, Haemophilus influenza, Moraxella, or Neisseria gonorrhoeae, or Chlamydia trachomatis are specific forms that result from sexual activity, okay? So these are actually venereal diseases. Okay, but these are all bacteria. Okay. A wide variety of bacteria, fungi, and protozoa can contaminate your contact lens and your lens case, and you can transfer them to your eye. You're sticking your finger in your eye when you put the contact lens in, right? Viral conjunctivitis can be caused by adenoviruses, but there are other viruses out there. Both types of conjunctivitis can be transmitted by direct contact and are highly contagious, right? Why is that? Well, you've got conjunctivitis, you rub your eye, you touch anything, you leave that stuff on a surface, and then somebody else can touch that and do the same thing, and then they pick it up, okay? Prevention, treatment of non-neonatal cases. Ciprofloxacin is for the treatment of bacterial infections, okay? It covers all possible microbes, bacterial microbes. Physicians may provide a prophylactic antibiotic eye drop even if you have a viral case in order to avoid secondary infection. If the symptoms don't improve in a couple of days, then we may have to re-diagnose. But you can see here the different types, right? These two, the causative agents are bacteria. Right? So we treat them with antibiotics. Okay. Here we can treat with oral and topical. And here we treat with just topical. Okay. This is caused by a virus. Okay. So what are the treatments? None. Okay. But we can give antibiotics to avoid secondary infection. Okay. Keratitis is an invasion of the deeper eye tissue, can cause blindness. Okay. Herpes simplex 1 or 2 cause it. An example of this is reactivation of herpes simplex 1, where the virus travels to the ophthalmic as opposed to the mandibular branch of the trigeminal nerve. Herpes simplex 1 can remain latent in nerve cells, right? So it can wake up and then travel down the wrong branch, okay? Herpes simplex 2, which can result from a sexual encounter or transfer from the genital to the eye area, all right, is another type of keratitis, right? So we can do clinical diagnosis, we can do PCR, DNA analysis, all right? The treatment is trifluridine or acyclovir or both, right? So what are these? These are antivirals. Okay. So this is a case where we have an antiviral on hand to treat you after infection. In acanthamoeba, the amoeba causes the keratitis. Now, acanthamoeba is a protozoan. Okay. So this lives in tap water and fresh water and is associated with poor contact lens hygiene or eye trauma, all right? So we would treat this with antiprotozoals, right? So you can see here, herpes simplex virus, and then the treatment is actually here we have antivirals. Okay. And then for something like acanthamoeba, we would use, say, antiprotozoals. Okay. My miscellaneous microorganisms, we're talking primarily about protozoa. What are the weapons? Here, latency, meaning it can take a nap. Here, there's a variety of weapons, okay? They can attack and avoid the immune system. So here are the diseases that we talked about, where they happen, what causes them, okay? This is a nice chart to sort of study, use as reference. Okay. So I'd make sure and maybe make a copy of that and keep it handy. Okay. And you can see the color coding here, right? If it's in blue, it's a fungus. Okay. If it's in brown, it's a bacteria. Okay. And if it's in green, it's a virus. Okay. And if it's in red, it is a protozoan. Okay. So there you go. This is what causes it, what the disease is, and what the pathogen is, right? What type of organism it is, right? So what you're seeing here is the possibility of any of these, with the exception of there don't seem to be any helmets on here. Okay. So here you're either going to see viruses, bacteria, protozoa. And fungi. Different entities, different types of pathogen. Remember that the virus is non-living. Okay. The protozoa is eukaryotic, as is the fungi. Right. The bacteria is prokaryotic. All right. So different organisms require different types of treatments, right? Most viral infections are going to be dealt with using immunization before you get infected, right? Whereas the other types we can treat during the infection. Okay. All right. So that brings to the end what I wanted to talk about today. Let me bump out here. Right. Start that. So we'll pick up chapter 17 in lab. Okay. And today I'm going to send you in lab a couple of things. If we go back to the PDF here, okay, you'll see that don't say that. Right? Okay. You'll see that we have an enteric bacterial ID lab, meaning what? We want to look at bacteria that come from the gut. All right. We want to find out which one of them are anaerobic, meaning don't need oxygen to grow, and which one of them are aerobic. Okay. So I'm going to send you that in your email. There'll be a form that you'll fill out and a resource that you'll look at. And then I'm also going to send you something from last week that you should do, which was the water pollution lab. It was I had to find an activity in a website to go with that, in addition to the microscope thing that you did on Connect. And I will send that along with the lab for the enteric bacterial ID. Okay. And then in chapter 17, that we'll talk about in lab, we're going to deal with diseases of the nervous system. Okay. So that'll be an interesting set of diseases and symptoms. Okay. Remember that the timed exam for 16 and 17 isn't going to be due until next Wednesday. All right. And make sure and keep up with your Learn Smart homework on Connect as well. And that'll help you with the material in the chapter. And of course, remember that Friday from 5 to 7 is office hours for anything you've got a question about. Okay. So be sure and stop in if there's something that's particularly confusing to you. And we'll go over that at that time. Or if you want to make a special time for me that doesn't work, if you have a problem with Friday 5 to 7, let me know and I'll set something up for you. And then we'll kind of gather together and discuss the homework and the exams and the labs. if you have questions about those. Okay. So any questions before we, okay. Jabus, yes, what is your question?
Yes. I don't know if I missed it or not, but our lab homework, is that being put on
That's good.
online as well?
That's, well, this week it was. This last
Okay.
week there was an online microscope exercise in Learn Smart, but this
Okay.
week it's going to be all stuff that I'm going to email to you. Okay, and there'll be a set of
Okay.
instructions that come with that, and then you just complete the activity and email it back to me.
Okay.
Yeah,
Thank you.
yeah. Okay.
I have a question.
Yes, ma'am.
With the Kirby lab that we did, were we supposed to get a lab report to because when I did the measurement, it said from the lab reports you gave us, we should answer the questions on it, and I didn't see no lab report.
Okay, there should have been a PDF that came with that, with the Kirby lab in your email. And what it does, what it has is just has blanks there for you to fill in the values you got from doing the exercise. And then from that, you answer the questions.
Can you please resend?
I
didn't see.
Okay, I can resend it. I can resend it. Yeah.
Okay, how about the grant same? Did you get my work?
Yes, I did.
Okay, thank you.
Yeah. Any other questions?
Yes. Are we having a midterm for you? What's our midterm for you?
The midterm is going to be next week.
Okay, so it's next.
That's
okay.
a lab midterm. And then let's see here. The lab midterm, and there's a lecture midterm. Let's see. Where would it be? Yeah, there's a lecture midterm. I'll tell you when the lecture midterm is when we get together for lab. I know that I put one in there.
Okay.
So I'll tell you where that is and when that is. And then I'm going to put up practice midterms to go with that as well. Okay, so that you'll be able to run through those and see how you do, and then take the real deal after you've done all your studying. Okay. All right. Well, thanks, guys. I'll see you in lab.
Okay. See you later.
Yeah. Meeting for